Ms. Middleton, a few questions from some of the court participants that I've been invited to ask you. Okay. This means we're going to jump rather from one topic to another. Okay. Starting with your time still in Scotland, and the reference that you made to a colleague sadly dying of hepatitis B, were any changes made to the way that you operated in response to that death?
Immediately, yes, we did become much more aware, I think, of the potential problems with the plasma we were dealing with. The person that had the accident, it was a known infected patient that caused the problem there. And that was in a lab, which was a separate lab where they were doing studies with hepatitis virus. But as far as PFC was concerned, obviously I suppose we did become much more aware of the potential.
Do I understand from that answer that the colleague was somebody who worked within the Edinburgh Royal Infirmary rather than within the PFC?
Yeah, she wasn't in the PFC, she was in the Royal Infirmary, but in the same sort of department, blood transfusion.
Do you know if the accident and the death was notified to any public health body?
I'm sure it was. It was a prick, it was an accidental prick with a needle, which is, tiny, but yeah, that's what happened.
Staying with your time in Scotland and moving to Glasgow, do you recall if the blood that you were dealing with, which was taken from patients with haemophilia, was marked as being high risk?
I don't think it was.
Do you remember if any particular procedures were in place for how you dealt with and handled that blood?
No, I don't think so.
I'm going to, this is a document that you haven't seen, I don't think is on our system, so I'm just going to read a short passage to you. It comes from a meeting in May 1985, a meeting in Scotland of the Scottish National Blood Transfusion Service. And it refers to a request that has come from you to the SMBTS. Forgive me, I've just lost my place temporarily. What is recorded in the minutes is this. You have asked, quotes, to obtain from the SMBTS about 50 litres per annum of fresh frozen plasma from which to develop a range of reagent biodepleted plasmas. You had approached SMBTS because of concern that the haemophilic plasma substrates in use at present might be contaminated with HDLV3 or hepatitis.
Yeah.
First of all, do you have any recollection of making such a request?
I don't, but I do remember the project. So.
Could you explain what that project was?
The project was to use monoclonal antibodies, the ones developed at the Royal Free, to make a factor VIII deficient plasma. Factor VIII deficient plasmas from haemophiliacs were used as reagents to test for the potency of a factor VIII concentrate. So rather than use these, obviously became much more potentially risky, that was thought to be the case, as reagents, and therefore we decided to use normal plasma and deplete it of factor VIII using monoclonal antibody column to deplete it.
In order to create assays?
To use for assays.
So everybody has for reference, it's PRSE 304075, and the minutes record that it was considered not appropriate to provide the material because there
Right.
was no surplus at the time.
Right.
Do you know if you were able to obtain an equivalent material from elsewhere?
I did get some from somewhere. I bought some from somewhere, but it wasn't the United States. I'm not quite sure. It can't have been English, British. I don't know where
Because
I
it was
got
commercial.
from. It must have been commercial.
And your desire was to avoid the material coming from the United States because of the perceived risk of HDLV3?
Well, it was to come from there was no point in doing it unless it came from a clean source. And I can't recall now where it did come from, but we did use that technology, and subsequently it was the assay was developed and was sold commercially by an organisation called Diagnostic Reagents, who are based in Oxford and who supplied, and probably still do, reagents for coagulation factors.
Moving on to a different topic. Was there any discussion about proposed research concerning AIDS, HIV, HDLV3 at Spaywood during your time there?
Not AIDS, no. I think AIDS and HIV were only really started to be identified in 82, 83.
But Spaywood wasn't involved in any
No,
specific projects to try
No.
to address the risk of
no.
HIV, HDLV3,
No.
etc. If you'll give me one moment. Further question. How much was your work either at the PFC or at Spaywood informed by a knowledge of the different severities of haemophilia in patients, mild, moderate and severe haemophilia?
I'm not sure I understand the question. We were making concentrates to treat haemophilia, whatever the level, however severe or mild it was.
Were you aware of the distinctions between
Yes.
mild, moderate and severe?
Yes.
Does it come back to the fact that your role as a scientist was to seek to make the product and
Yes.
not to decide how it should be used by the clinician and the patient?
Yes. Yes. Although in discussions with clinicians, because we were very close to the clinical use, particularly in the UK, you did get to learn about what people were doing, what clinicians were doing for treatment, etc. So
those are the questions that we've had from the CP. So I turn to you, sir, now, to you have any further.
Well, I just have a couple of questions. You've described life in the basement in the old Royal Infirmary in Lauriston Place, which I can imagine was like working in a basement. But you moved to Middleton.
yes.
Did you? That would be about 1973, I think.
Possibly, yes.
What was life like there?
Oh, well, that was very different because it was a purpose-built facility.
So you had a purpose-built facility
Yes.
there, and then when you moved down south to Wrexham,
Yes,
you found yourself in a porter cabin in a
and a garage.
garage.
I only did the research work in the garage, I should say.
So would I be right in thinking that David Heath, whose enthusiasm had recruited you to the post,
Yeah.
he would have had some schemes and dreams for improving those facilities?
Well, he did, ultimately. They did improve that facility, and they had a purpose-built production facility for high HC. on the Wrexham estate.
So you did eventually get the facilities you wanted for research as well?
Well, it didn't happen until, I'm not even sure the timing of it, but it was 82, 83. I mean, it was later than I was. All my work was done in the garage and the porter cabin. Under laminar flow, I should say. We did clean up the porter cabin.
Did you know, this is a second topic, did you know anything about the Spraywoods continuing to market Coate without, it may be thought, telling Coate they were doing it by buying it and sourcing it from
I didn't.
an intermediary?
No, I didn't. And
is that the sort of thing that David Heath might do, as you would see him?
well, I suppose, I guess he might have done. I'm sorry to say.
I mean, you have a slightly, I suspect, a slightly ambivalent attitude towards him.
Well, he was the sort of person that I had two mentors. One was Jim Smith, and one was David Heath, really, on the business side. But I didn't, but only in the sense that it was all his ideas and everything else that I admired. But when it came to he did do certain things that I thought were a bit near the edge, and it wasn't my style at all, and I didn't really like it. And it was very unfortunate because it caused problems with BPL, which I could see why, and my two mentors didn't get on at all, Jim Smith, and because they were opposite ends of the spectrum. So it caused difficulties.
Yes.
So I'm afraid it was the downside. The upside being he had these amazing ideas, and some of the ideas like putting polyelectrolyte into a donor centre to collect plasma straight from the donor onto the polyelectrolyte and then fractionating it on site, as it were. I mean, it's a very interesting idea, but obviously plasma fractionation was not going to carry on in the same way it was never going to happen in the future. But had plasma fractionation gone on for, had recombinant technology not started to take over, then it would have, it was a really good idea. So he did have very good ideas, but sometimes bringing them to fruition was not quite so good.
Yes, was it a case of the next big thing, as it were?
A little bit, yes, I'm afraid so.
Yes, well, thank you. That's all that I have to ask.
Okay.
Ms. Middleton, with all of our witnesses, we ask at the end if there is anything else that you wish to say in your evidence.
No, I don't think so. Thank you.
Thanks.
Well, we've asked you a number of difficult questions this afternoon and detained you, I think, for rather longer than you might have hoped this afternoon. I'm sorry about that.
That's all right.
But I'm very grateful. And you've done two in particular difficult things, it seems to me. You've explained to us how the polyelectrolyte system works in terms which I think I can understand, and those around me will. And you've answered questions which you didn't really want to have to answer, but have done so, and done so in order to help us. And can I just tell you that's very much appreciated. And thank you for coming the way you have down here, and I hope you have a good trip back tonight.
Thank you.
Mr. Hill, what is next week going to hold in store for us?
On Tuesday, so we will return to the presentations. We will complete the presentation on Spaywood, and then we will move on to the last of the current set of presentations on pharmaceutical companies, which is on Alpha and associated companies. Then later in the week, we will move on to some presentations about haemophilia centres.
Very well. So 10 o'clock on Monday. 10 o'clock.
So I think Tuesday.
Oh. I've done it again. Sorry,
Unless.
I do apologise. it'll be 10 o'clock for me, or before 10 o'clock for me on Monday, but for you, 10 o'clock on Tuesday. For us, 10 o'clock on Tuesday.